Yeah. So can you hear me all right? I guess you can see. Yep, that's good. You find the recording's all right? Sorry. All right, so what we'll do today is just finish off the last few slides, and then we'll go into a revision mode. So have your questions ready, or I'll try and get some questions for you. I did try and put a Kahoot together, but it didn't quite work. But anyway, we'll just work with what we've got with, isn't it? All right, just to remind you, tomorrow is the deadline to say if you want to do a take-home exam or do a face, you don't have to say you want to do a face-to-face, just have to contact me if you want to do a take-home exam. Like I keep going on about, so we release the instructions on the 18th, submission by the 21st, and the oral exam is the day after. And just to also confirm that the materials covered are those which weren't covered in the midterm. So it's easier to say, I suppose, there won't be the introduction, metabolism, vinegar, kombucha, and wine. So they won't be in it, but everything else will be. And yes, there will be a question on Shane's sensory. I can assure you of that. And I don't think we had enough of that. Yeah. So is there any questions about that? All right, that's good. All right then. So and oh yeah, just make sure you do the quizzes before tomorrow night as well, because then I don't think I shut them down, but anyway, it's good to get the quizzes done before you do. And I can't, and I think obviously the 3260 people have lab reports. And yeah, I suppose I should talk about those 3260 people. Don't worry if you're 3270. For the 3260 people, I heard the kombucha lab didn't quite go as smoothly as it should have done. So what I've done is I've uploaded from 2019, when we actually were back in the lab, the sensory analysis and all of the acetic acid analysis. So they did do ethanol at that time. So to complete the report for the kombucha, which is only, I think, about 3 questions, you can use that data that's posted on the course link. Good. Oh yeah, will you be releasing the answers to the quizzes after they close? Well, I could do, if you want me to, I can do that. Usually I'll do a few attempts until you get them all right, but I'll release it. I've just got to make sure, obviously, everyone's done the quiz, because I can't release it when only if somebody hasn't done, because they'll, should we say, take advantage of a situation. But yeah, I'll try and do that if I can remember how to do it. Right, I think I can. All right then, so when we left it on Tuesday, we're talking about strain development and strain isolation and improvement. As we talked about, when we get a new isolate, it's not domesticated. So what we have to do is domesticate it, become more stress tolerant, to be more productive, to utilize substrates and not produce byproducts, and maximize the yield of whatever we're producing, be it a glutamic acid, antibiotic, or whatever it is. So we can get the isolates from a specific niche, and people can't, they don't do as much as they used to. Like I said, back in the 50s, as a microbiologist, you spend most of your time in the field, in a jungle or something like that. But these days, what we really rely on, isn't it, is genetic modification. So in the olden days, in the 60s and 70s, what you would, or even in the 50s, you would mutate either by natural selection, getting successions of population and selecting those that give you the desired product, like high antibiotic production, or you use UV light, a mutagenic agent, to help nature along, which has the same effect, but it became very non-specific. So you're not just mutating one gene with UV and aquadine orange, things like that. You're obviously affecting quite a few. And so things advanced, and then in the 70s, they started to bring in site-directed mutagenesis, which was a bit more specific, very laborious. You have to kind of get the gene out, to melt it, put the primer in. You need to know the sequence. And back in the 80s and 90s, getting a whole genome was very difficult. Even, it took about 4 years to get a bacterial one. It took 13 years, I think it was, to get the, maybe 11 years to get the human one. But when we get the enabling technologies, DNA sequencing, DNA synthesizing, things really sort of took off then. And we talked about how we can genetically modify organisms by inserting genes. We use our restriction enzymes, ligate it to a vector, place it in, transduction, transformational conjugation, and that expresses in the cell, and we're being very useful for that in terms of understanding form and function, but also genetically modifying certain strains. So the professor before me, or before him, developed a yeast strain which had the malolactic fermentation in the yeast itself. So it did the wine fermentation and also the malolactic. It didn't depend on Oniococcus. And you would have thought it would have been successful. Well, I think he gets about $5 a year from it, so it's not very popular. And we'll talk about why it's not popular in a little bit. But then we came to the game changer, what people are suggesting is a game changer. Everyone's a game changer, isn't it? This one to say is, because if you can certainly be specific in your gene editing, that's a bonus. It's obviously not affecting anything else, you're specifically affecting that. And rather than just doing one base at a time, as in, basically one amino acid at a time, why can't we just insert whole pieces of DNA, whole genes in a specific place? And why can't we knock out specific genes? So when we talked about lysine production, we knock out homolysine dehydrogenase. Well, that took a bit of effort, but why can't we just edit it? Anyway, so long story short, and I think I covered this last time, but we'll go through it a little bit, is CRISPR was discovered. So as we talked about last time, CRISPR is clustered, meaning it's together on the genome, regular interspaced, so it's got these repeating units with a spacer in the middle of them, and they're repeating. So as I mentioned to you, essentially what we have, these pieces of DNA with a space, oh, come on, pen. You always got it down. Oh, the pen's gone. There you go. Well, you got the, oh, there you go, that's better. So you've got these palindromic sequences, and these are the spacers. And as we talked about, and I'll tell you the story about CRISPR in a little bit, these are phage DNA, and what the cell uses them for is an immune system against phage attack. So what happens is your phage comes along, you're wanting to make you a victim because you're a bacteria, injects its DNA, and as we talked about, it can go to lysogenic or it can go into a lytic cycle. We'll talk about lysogens for now, it's easier. And so what happens is that if the cell's been exposed to that phage before, it starts expressing what we call the this spacer, this sort of unit here, which has the DNA from previous infection, which is passed on from generation to generation, and also the palindromic sequences. Now, what happens is the palindromic sequences read the same forwards as they do backwards, and so they naturally form this loop structure, the self-hybridized, and that's called the guide DNA. And now what happens after that, so we'll talk about it in the next slide, is the Cas9 protein, in the case of Streptococcus, is a protein that has helicase activity, i.e. unzips the DNA, and it's got nuclease activity, which cuts the DNA. So if you like it, zip open, cut, and then leave it to be. And obviously for the phage, that's pretty lethal. For the simple fact, you've cut the DNA in half. So that's how it works. And as I mentioned, it was a big discovery at the time. And what people use it for is for gene editing. So we know the palindromic sequence, so we can have that, but we don't need phage DNA here. We could put any sort of RNA, couldn't we? We could hybridize it to a target, because we know about DNA sequencing, because we've got machines doing it all the time. So in the bacterial world, it's important for phage protection, and that's why phage mutates so long, because they have to change in order to try and avoid these systems, the sort of arms race, if you want to call it that. But when we come to eukaryotics, or even prokaryotics, what we can do now is just do what I've just said. It's that we can cut a piece of DNA, which inactivates the gene, or we could insert a gene of interest at a specific point. And that you can't do that with directed mutagenesis. You can do it, I suppose, in a way with all these other sort of techniques, but it's very difficult. This is very specific. So is everyone good about that? Good. All right then. So I like this story. I don't know why, but maybe because it's a lesson of academics. So do you know who discovered Caspar then? Or CRISPR, not Caspar. CRISPR. Caspar's a ghost, isn't he? No. All right. So Jennifer Doudna was a scientist, I think was in Berkeley, something like that. Back in 2006, she was sequencing E. coli. And whilst she was sequencing E. coli, she noticed there was a lot of sequences that didn't account for anything. And more than that, there was these sequences that were palindromic, as in they seemed to go palindromic sequence, about 40 base pairs, spacer, and then another palindromic sequence. So she said, oh, yes, a bit odd, isn't it? And then but can you think what we could use that? Well, this is an unfair question. Do you know what we used that for back in the 90s when we saw this sort of, these sequences where there were palindromic, spacer, palindromic? It's obviously a question. You can answer online. All right, so we used them for DNA typing because within those spaces, it was variable. So you could say, well, if this strain has this DNA fingerprint, because we used to use PCR to express it, we got a DNA fingerprint. So we thought, oh, that's good. We can differentiate closely related strains based on their DNA fingerprinting and these intergenic spaces. But Jennifer did, no, Dr. Downdu did something else. She saw this and she said, it's very odd. Why would this happen? What's even 30% of the E. coli genome was contained this so-called junk DNA. Ours contained junk DNA, but that's another story. So she went to a conference in about 2008, I think it was. And as they're doing conferences, we present data, go to the expo, go to the free bar if there is one. But there was a scientist there who said, I recognize those sequences as phage DNA. I thought, that's good, and that's why it's good to go to conferences. You learn these things, apart from the freebies and the expo. So she said, that's good. That's very surprising. Now, don't ask me why they knew phage DNA so well, but they did. Now, she went to another conference and presented this, saying, look, we've got this genome, we've got the palindromic sequence, we've got this phage DNA. What could be going on? And this is where she met, say, Emmanuel Carpentier. So Emmanuel Carpentier was a biological structural scientist, biochemist, I think a structural biologist, that's what they call them. And they collaborated together because that's what's another good thing about collaborating, different expertise. And this is where they discovered the Cas9 system, i.e. what I've just talked about in CRISPR. So that was good. And now what they did, as academics do, is they publish papers. Because if you don't publish, you perish. That's the old term. It's probably true as well, up to a point. So they published this paper, and they also thought this could be very useful for gene editing. They knew the potential of it. Unlike other sort of people who discovered things which, if we don't go to Twort, for example, in this phage, he didn't see the value of phage, whereas D'Hillier did. Anyway, so what's happened is that Downder and Carpentier said this could be a good mechanism to get a phage-resistant strain to do a yogurt fermentation or any dairy fermentation. So they opened a company up to produce this technology, a sort of phage insensitive or phage-resistant starter culture. And also, as good scientists do, they put a patent in saying, this patent about gene editing. Now, in that patent, they actually did have a part which was saying about eukaryotic cells. They worked with only prokaryotics, but they said we could also use this for eukaryotic cells to solve or cure gene disorders. Now, the problem was back in that time, which was about, oh God, my dates, 2012, I would say, about there, maybe 2013. Back in that time, you have to have experimental data to support any patent claim. Now, these days you don't. So you could go and make a patent and you could say the astounding things. Now, whether the patent lawyer will accept it, because the people dispute it, that's another matter. And the patent people said, well, no, we can't give you a patent for the eukaryotic cells because you've got no data. You only got it on prokaryotics. So in comes George Church. So George Church was from MIT, and he read the paper that Dowder and Carpentier had written. And he was a genetic engineer, basically medical, clinical. And he said, wow, this is a good method for doing gene editing. This could be really big, in the medical field, a whole new field where we could cure genetic disorders, which otherwise wouldn't have happened. Now, George has a postdoc called Feng Zhang. So Feng Zhang was working with his George, who's his boss, and Feng was a bit of an entrepreneur, so he actually took the results he generated under poor George's supervision, and he submitted the patent for eukaryotic sort of gene editing using CRISPR. Now, obviously, this upset quite a few people. It didn't say it upset Dowder and Carpentier because they failed to get this patent in on time. But Feng was very good. He said, "Oh, I've got the patent now," and there was a litigation war. So this litigation war raged on. It still does. There was a big decision last month, but I'm going to ask you a question now. It's not the odd one out, but who won the patent war? Was it George? Because George said, "Hang on a minute, that's my lab, and I funded you for that." Was it Feng, the postdoc, who kind of stole, if that's the best term, stole the IP, or was it Jennifer Emmanuel who actually first discovered the system? Who do you think won that patent war? You can answer online. I guess. Oh, poor George. No, well, what happened in the end was, I hope the two women, well, you could hope. So the reality is, what happened was that Daunder and Carpentier won Nobel Prizes with, and I don't think they split the money. I think they're both, well, they could have split, I don't know, but they got famous. Now, what happened is that they actually came on the side of Feng. They said, "Nope, you're right. You submitted the patent first. We accepted it first." And the trouble was, though, it's taken 4 years, and I think it's cost him $30 million. And the problem is that the US system recognizes patent, but what happened to Feng is that he had a co-inventory brought on board to have the patent, but then he threw him off the patent. And in Europe, if you throw someone off a patent, it instantly becomes invalid. So basically, he lost the European patent. He got the American one, but in this meantime, since there have been raging wars, other companies have found loopholes. So there's more than one Cas9 system. There's lots of different systems, and the big companies like Millipore and a few others have established, and and when you're doing patents, you get loopholes, and that's the thing about patents. You can't have them so broad to cover everything, but you have to make them specific. The more specific you make them, obviously, the more loopholes and ways around you can get. So the sad situation is that, well, it's not sad. Daunder and Carpentier, world famous, wealthy, they don't need to worry about grant money. They're always getting it. I suppose Zhang is a bit happier. He's got his company and people, but nobody's actually used it commercially. They've demonstrated it. They've demonstrated, we can cure sickle cell anemia. We can cure blindness and things like that. But poor George, he came out with nothing. But George is one of those typical academics. So what he's going into now is trying to revive mammoths using his gene technology. And I think he also got involved with personal genome sequencing. Basically, people can just take their own genome. So it's like a true academic. He kind of follows things without making money. But it's an interesting story because there's a few lessons, I suppose, we could learn. What lessons do you think you could learn from this experience? Well, I think the first thing is that when you're at conferences, you've got to be fairly careful what you say and who you talk to, because it literally is. Some people take your ideas and squander away and use them for themselves. I think it just illustrates, this sort of dilemma people have in that you get a new technology and you want to tell the world, basically it's like Banting with insulin. If Banting and his insulin behaved like Zhang, it would have been a bit bad. Although it's a colourful story, isn't it? The insulin one. So you've got to patient protect things, and you've got to know the value of things in terms of what would it mean in the future. And just be careful who you get as a grad student and postdoc, I suppose. So that's the last lesson. But anyway, it's an interesting story. I like that one. Anyway, I'll go on with it because it's. So obviously, people, when they think about CRISPR, they talk a lot about gene editing and curing sickle cell because you do it in living cells. You can literally cause muscular sclerosis. And in microbiology, we can go down what Doudou and Carpentier initially thought, saying, well, it's a really good way of getting phage-resistant strains. And that's important, isn't it, when you're looking at starter cultures? Well, we can do gene editing, making our sort of strains, so we cut the branches off, making enzymes not sensitive to feedback inhibition. We can take out virulence factors much easier than we can do today. And also, we might be able to insert genes to make synthetic drugs, but as I mentioned, that's not as easy as you say. But I think the big thing people talk about when talking about genetic manipulation is synthetic life. And, when we talked in the first unit about the alchemists, the alchemists wanted their philosopher's stone and they're creating life. And if you remember back to Twartz, he left phage to try and make artificial life in a test tube using electric field, electric arcs and this sort of soup of components. And I think he made an amino acid and say, oh, look, this is evidence of life. But, back then it was just, it's just what it is. It wouldn't have occurred. It wouldn't have ever worked. But then people started obviously thinking, well, if we're going to make artificial life, you have to start with the blueprint of life. You can't start with the products, you start with the blueprint, and the blueprint is obviously genetic coding. And so the thought is that you could just artificially make a genome, because we've got DNA sequencing where we know we can get the sequence, and we've got synthesizers where we can do long strands of DNA and print them, and we can stick them together, put them in a cell and spark it into life. So the concept of synthetic biology you can date it back to 1970s, but that time was just all virtual, it's concepts. And then in the 80s and 90s, and even now, people do it virtually. But they are getting real systems. And one of the big advances, if you call it an advance, is the BioBrick system. So what the cell is, essentially, isn't it? There's two things on it. There's the DNA genome, which is the fingerprint, like the telephone directory, if you want to call it that. But then you have how genes are controlled. So it's no good just having genes there, but how do you control them? And what the BioBricks were is this standardized system for making synthetic genes. And what we mean by synthetic genes is that when we look at gene regulation, we know we've got promoters, i.e., where does the DNA polymerase land in order to start transcribing the DNA to make messenger RNA. We have terminators, which tells the DNA polymerase to stop. We have repressors, we talked about those. We've got promoters, we've got parts which are ribosome binding to initiate synthesis. And also we have what we call reporters, but that's the gene of interest you're putting in. Now, this is what, and you can go online if you want and see this is a sort of public charity sort of thing. You go online and you can see a database of all these different sequences, which are either promoters, terminators. And what synthetic biologists do is they use these promoters to put them in a virtual genome in order to express a certain enzyme. And you might think, well, it's easy, but what it's done is taken a lot of the sort of workload off designing things from zero. It's like a library you can go to just to get, oh, this is a promoter, this is a terminator. Because what tends to happen is that they thought, well, we could just put these genes together, and once we put the genes together, they'll start working. But as we've learned, isn't it, in our sort of unit 1 and beyond, everything's interrelated. It's a circuit. Certain genes have to be on at certain times, off at other times, then upregulated, downregulated. And in a lot of ways, it's like an orchestra. And these sort of DNA sequences, and even if you want to call them BioBricks, they're basically an orchestra. So you've got the violins and the drums and the cellos and whatnot, but they're nothing without a conductor. And that's the problem we have today. There's no real conductor to coordinate these things. So when we talk about artificial life, it's much more complex, even to do the most rudimentary sort of biological reaction. So there was some work done on bacteriophage infection, essentially a lysogenic phage which infects the cell, and it decides, does it go to lysogen or does it go to a lytic cycle? And they drew the network for that, i.e. the decision points, what proteins interact with other proteins. And it was so complex, and that's just that decision. So if you think about a metabolic pathway, it's much more complex, isn't it? So the thing is that BioBricks has kind of helped because it's got this sort of, if you want to call it a Lego factory, you can do, pieces of Lego where you can go in, get a promoter, get a repressor, put them together and put your gene in and make a cell. And they do it experimentally now to verify it. But if you remember, I think it was Tim Knight who did this, they claim saying, oh, we've made the first artificial synthetic bacteria, the mycoplasma. And the reason why they took this ruminant bacterium, or the bacterium found in the rumen, it had the smallest genome, 1 million base pairs. And they didn't really make synthetic life. What they actually did is they sequenced it. They then stuck it all together with this, by synthesizing the genome, and put it into a living cell. And that living cell then duplicates, and the cell line obviously went for the artificial DNA, if that's what you want to call it. So the fact is, though, they can't kickstart it. They can't kickstart the cell from ground zero. And regulatory-wise, they've just basically copied a living cell. They haven't really done anything different. So if you say synthetic life's down the corner, I think it's a few corners away. But they've done it to yeast as well. Instead of expanding the number of things they can do, which I suppose in some ways an improvement, because at least you can kind of put genes in you want to put in. But this sort of feeling that you're going to have brand new life, I think well, it could happen. It's not in my lifetime, but who knows what happens in the future. But there are ethical situations about this. All you have to talk about is COVID and man-manufactured. But it is there, isn't it? You can imagine somebody in a lab saying, oh, I could get the most virulent virus or virulent pathogen, and it won't be detected because it's going to be a brand new organism. And I think this is the ethical side of it. And I suppose in some ways why CRISPR might not get adopted as much as it could be. But after saying that, you can see so many benefits to CRISPR in terms of curing diseases. I think the benefits outweigh the thing. But, when ethics gets involved, it's all a bit dodgy, isn't it? Anyway, so long story short, though, like I say, that's way in the future. Well, you can make the future. The expression systems we use, as I said, the E. coli was the kind of workhorse. And in some ways, it kind of restricted what we could express in terms of proteins, and high GC content genes couldn't really be expressed that well, and you can't do the post-transcriptional modifications as you would do in a eukaryotic cell. So what this is leading to now, and because we're understanding much more about genomics, and we're basically decoding many organisms, about God knows how many hundreds of thousands. We're going to other eukaryotic host cells because they can do this glycosylation, putting proteins or sugars onto proteins, which sometimes they have to do. And so currently we are following the synthetic life, but a lot of it's to do with trying to find alternative hosts to express these modified proteins. And as I mentioned to you, a professor before the last one, he spent a lot of his academic career developing yeast strains which could do male active fermentation and things like that. But anything with GMO isn't really in for the food industry, even if it gives benefits to it. Certainly in the production of amino acids and other sort of valuable products, you could see the benefit of GMO there, but in food fermentations, it's all back to the past and trying to recreate them. But you are getting to the point where, we will be able to genetically modify to produce those products that we're going to meet the energy of the biobutanols or biofuels. New drugs, it's interesting that drug development is more chemistry these days, which is a kind of throwback to the olden days on biological, because, we can all produce things chemically. But, always going onwards and upwards to more performance and things like that. And seeing this sort of interaction between microbes, the microbiome and other sort of applications, that's where the future lies. Anyway, that's that. All right, so there you go. That's the story and a half, isn't it? So what I can do now is I can turn it over to yourselves, and you can tell me if there's anything you want to go over, or if you, like I say, I can think of some questions, but I'll answer your questions first. Oh.
The GMO so opposed in the food industry when there's like GMO corn and stuff that's used for eating?
Yeah, it's a very good question. So, why are we reluctant to do food fermentations when we have so much GMO corn? And it's very true. If we didn't have GMO corn at the moment, we would be really seriously stuck because of the weather we've had and obviously the restrictions of world events. So in the industry, it's as I say, it's very traditional. It's you're almost like going back in time with some of these, and they make a big emphasis saying no GMO, no preservatives, even though you don't need preservatives. But certainly in the wine industry, as I said, this is a life's work almost of this poor professor. And they just said, well, no, we'll rather stick with the traditionals. And I think that's what food fermentations are. But certainly in the field of citric acid production, biofuel production, they're really open to the genetic manipulation. Is anything specific then, or? All right. I don't know what to. I'll go from brewing, and we'll see how far you want to go. But just tell me if you've. So thanks a lot, but we're going home now. Yeah. Do you want to go over any? Well, I suppose I should be asking you. Do you want to go over anything, or you don't want to? You're good.
I had a question about, I don't know which you asked it was, how does the glycols like shouldn't work in that one? I think it's glutamate. Is that glycolate shunt in PCA? How does that work? Like, does it replace the glutamate that?
Which one was it?
I think it's in the amino acid glutamate production.
Oh, glutamic acid production. Yeah. All right. Oh, the anaplerotic reaction. All right. Let's have a look. All right then. So so this is it, isn't it? So just to go back a little step, I won't go through all of it, but it's important to see where it comes from. So glutamic acid production, which you. Oh yeah. Oh, there you go. Good idea. So with glutamic acid production, then we know about this reliance. Well, we'll do this first. So it's all to do with biotin deficiency, as in the biotin catalyzes is a cofactor for an enzyme that builds up fatty acids, which build up membranes. And what happens if it's biotin deficient? This enzyme that synthesizes these membranes isn't working efficiently, makes a leaky membrane. But we can get the same effect by using surfactants and antibiotics. So one of the key parts about this pathway is that we have to get the cell to overproduce glutamic acid. And we do that, as we talked about, by biotin deficiency, and it naturally produces it, just got it to get more. Now, any time something's taken out the Krebs cycle, it needs to be replaced in what we call anaplerotic reactions. And these anaplerotic reactions are literally Greek for replenishing. And these are the rate limiting enzymes, pyruvate carboxylase and phosphoenol pyruvate carboxylase. And so what these, here we go. So what these do is that they feed in at the point of oxaloacetate with obviously the malic acid combined with the acetyl-CoA, isn't it, I think? Something like that. Anyway, so what happens is that we've got non-growing conditions, because as we all know, we have to have non-growing to increase the productivity. And we deregulate the enzyme. We bring out the glutamic acid, and we'll talk about that on the next slide, how it happens to do that. And the rate limiting enzyme reactions, like I say, the pyruvate carboxylase. So by knowing the rate limiting enzyme, we increase its activity, through overexpression to produce it. So it's a very interesting sort of cascade reaction. So we've got our biotin here, and obviously biotin, as we talked about, is synthesized these fatty acids and membranes. But the cell senses that the membranes aren't good. There's something definitely going on here. And we can recreate that with surfactants and antibiotics like penicillin. But then the sensors are the serine protein kinases, and what they do is affect the 2-oxyglutarate dehydrogenase to positively, and this is a sort of complex of enzymes, which will take the 2-oxygluteric acid out of the Krebs cycle, produce glutamic acid, as we talked about, because of the deficiency in the membrane, it just leaks out where we can harvest it. But a key point is, as we've talked about, is this one, the pyruvate carboxylase and PEP carboxylase, which essentially causes, sustains the reaction without it growing. Was that all right, or?
Yeah, is any of this the oxalate shunt, or is that a separate?
No, the oxalated shunt, that's a good question. So what the cell has to do is two things. One, it cannot produce too much ATP, overheat radical burst. So the glycolytic shunt is essentially a means of keeping the Krebs cycle going without producing reducing equivalents to harness the electron transport chain. So really, it's what it's designed for is to make glucose. The glycolytic shunt is a anabolic. People impose it onto the Krebs cycle, but the actual fact is a totally different cycle. It's there to produce glucose from things like acetic acid and things like that. But what you can also use it for is to generate intermediates, because it is similar to, as we talked about, the Krebs cycle, except it's got a whole chunk missing. It goes through the glycolate shunt part to miss a lot of those reducing factors out. So it does participate for sure. And but the key, well, I keep saying it, but the key point is this sort of leaky membranes, this cascade reaction which activates the oxyglutarate dehydrogenase complex and the replenishing reactions. All right, so we'll go online now. So what are the main concepts we should have for each unit we should focus on? That's a good question. So, like I said, I know I keep saying it, but I'll say it again, is that all the material, there's like two levels, isn't there? There's the level of learning, saying the basics, and there's level of application. For example, in this last unit we did with Dowdy and Compartendia, I won't say a question saying, tell me the story about CRISPR and how George and Faye interfered with life. It's getting the concepts, and this is why the unit summaries are so vital, well, not vital, but it's an important learning aids, because they give those concepts. And as we talked about, and you just go back to the midterm exam, it's all to do with not knowing specific fact. Well, sometimes it is, but most of it is application. Now, having this sort of understanding of, well, for the glutamate production, you know, if I was setting an exam question, obviously you want to say, well, what's the microbe? What's the conditions we do for the fermentation, and what's the mechanism? Because they're key concepts of how we manipulate pathways and how we can upregulate certain enzymes to overproduce. But so when I say concepts, it's exactly what it is. And you know you're going to have multiple choice questions, which you're going, and you know you're going to have short answer questions, and you're going to have long answers. So it's exactly the same format as the midterm, except obviously covering much more material and the material not covered. All right. Yeah, so go to your unit summaries, and if you haven't made, we don't do unit summaries for all of them, but I would suggest, up to you, to make unit summaries for those ones that weren't set assignments, if that makes sense. Any tips for the take-home final exam? Yeah, so for the take-home exam, as it's the same format as the midterm again, where you have to develop questions. You know, basically prepare an exam of an exam. And I find it's very useful because when you're writing an exam, you think, wow, it's really difficult to answer these questions. But in actual fact, it's more difficult. I know you don't appreciate it at the time to actually make these questions, because questions have to be clear, but they also have to be in alignment with the learning outcomes. So as I said to a few of you, you know, when anybody develops a course at university, we're accredited. So accreditation is where an outside body comes in and says, yeah, this course is fit for this sort of standard. And part of the accreditation process is to come up with learning objectives. And a lot of accreditation bodies come up with their own, for better or for worse, usually the worst, but that's what they do. They give you saying, well, you have to teach them X, Y, and Z. Now, when it comes to say, well, how successful have we been at delivering the learning objectives? We say, well, we do exam questions, we do other things than that. So if you had to have a tip about take-home exams is, and I know I've said this to a few of you, look at the learning objectives. Yeah, basically, you know you're going to be tested on the learning objectives. So how would you put those in such a way in order to test not only what's in the learning objectives, but the sufficient level? So the only tip I can give you, and well, a few tips, read the learning objectives, make questions clear, make questions that aren't not specific fact-based, shall we say. Make them that test application of knowledge rather than facts, you know, about saying, well, I'll give you an example. I can't think of one at the moment, but I think you get the idea. Well, I think a lot of you did all right in the midterm who did it. But then the day after, we're going to do the oral exam where we'll ask you 4 or 5 questions and see how you answer those. So again, 70-30 split. And it's not easier than the sit down. You know, it's about the same. You know, in both cases, you need to know things in order to do it. All right. So, like I say, if you want me to go over anything, I can. If you want me to give you some questions, I can, but it's up to you. One silence. Well, it's only 2 of you. So what do you want to do? Do you want to call it a day now, or do you want to keep going, or up to you?
I can ask specific questions if you want.
Well, it's a few fun.
I don't know if you want to cover anything else. So.
Oh no, like I say, I'm here till the time, so don't worry about my time.
Concept of the fed batch culture in the context of biomass. You basically like grow up your microbe that reaches a biomass you want, and then you feed it just a bit growing. for yeast, if you want to grow it, it'll be like it's pretty high bricks, isn't it? Really,
Yeah.
under the factory effect, but when you're feeding it, you want it to be aerobically growing.
All right, so that's a good question. So what we're saying is that why do we start with a batch culture then go to a fed batch? Well, the initial batch culture is there just to get the biomass base, if you like, because if you could imagine starting with a very small volume of media, then start feeding it in, it would take so long to do because you're kind of depending on that. The thing about a fed batch culture, it doesn't make things grow quicker per se than a batch culture. But what it does do is make it more efficient. So you're exactly right what you said in that when you do a batch culture, everything's nice with the world. You've got so many nutrients there, you've got nice pH and probably everything else is good. And you're wasteful. Your Crabtree effect takes over if you're a yeast, making alcohol, ethanol rather than going down the TCA cycle. And your maximum growth rate, you're really going for it. But when you look at the growth yield, you say, well, put all this stuff in and I've only got this yield left. So with a fed batch culture, you start off with a biomass base, if you want to call it that, and then you start feeding it in at a controlled rate. So you don't do super fast, you don't do super slow, because we talked about if it's very slow, the maintenance energy takes a lot of that and uses it for non-growing functions. If it's too fast, you get slip reactions and all futile cycles going on. So we find the optimal growth and that enables this sort of coupling of anabolic and catabolic. And that's why we get much more biomass compared to when it's just a batch culture. And going on from that with a continuous culture, obviously with continuous culture, it's just hanging on in there. So, and it produces a very low biomass because we need to have low carbon of energy sources, because if we have too many, it won't be limiting, to be excess. Oh, yes. Is there any way we can see what questions we got wrong in the quizzes? Oh, yeah. So I thought we could do that by just retaking it again and again. Yeah, like I say, I'll release them when we get when everyone's done the quiz. Like I say, I can't release them whilst people are still doing the quiz, and there's quite a few people who still got to do the quiz. Anyway, yeah. All right, so I'll give you a question then, and you can tell me. I'll where we're going. All right, let's just go to the yeah, so I'll go through the units, and then, like I say, when you say, "Look, I'm off," yeah, well, I'll stop. But all right, so we'll start with brewing because, as I say, that's going to be the one that's. Oh, I'm just gonna. I'm going to give you some clues now, but anyway, you'll see. Like I haven't written the paper, I haven't actually written the exam, so I'll probably forget all this stuff. Blame me if yeah. Oh yeah, I suppose I should say, for those who are going to take the sit-down exam, I will be releasing a set of example questions on the 9th, which is Saturday, because like I mentioned to you, there's going to be a very firm cut-off point. After midnight on Friday, you're basically going to be doing the face-to-face. So on the 9th, I'll be releasing example questions, and again, these are the type of questions. These are not going to be the questions, but it'll give you some practice, I guess, on how to, what sort of questions to expect. All right. Oh, don't worry, I'm going to get there eventually. It's just. All right then. So, and you can join online on this one. So we talked a lot about the water, but what is the interrelationship between water and malt and hops? All right, I'll give it another question. So why is Dortmund lager, this one, darker? Whoop, yeah, why is Dortmund lager darker than Pilsner? And so that's, so I'll just go on to that. Right, fair enough. Yeah, so when people ask about what do we need to know, like I would expect you to know, say, well, you've got to tell me on average how many mgs per liter is of calcium would you get in Pilsner water. But the key concept is about pH and flavors. So if you remember, when we talked about Pilsner, for example, uh, it's basically low in everything, low in all the low in the minerals and low in bicarb and calcium. And so that's got no flavor. In actual fact, you have to add a few hops just to make it taste like something. But then we get to Dortmund, which was down the road, and lagering is a fermentation technique. Even though people, make it out saying, well, only blonde beers are lagers. It's not true. Lagering is a process rather than a beer style. It's not doesn't mean to say all lagers are the same. So in Dortmund, their water's harder for definite. And what we have, isn't it, is this sort of balance. And it's all to do with balancing rather than absolute amounts. So when we talk about pH, we know about pH because we know with pH, we have to hit the right pH for mashing. That golden corridor between 5.1 and 5.4 in order to make the enzymes work. And those enzymes are going to break down the starch, they're going to break down proteins. And to get that that sort of target pH, you say, oh, well, just add acid. I wish that sometimes to do, actually. But what the traditionalist does is look at the chemistry of calcium and bicarb. We know it's all about reducing the buffer capacity. The bicarbonate is our blood buffer, and blood buffer is obviously neutral pH. Pilsner is own calcium, which is less faint. That's true. Yeah, basically it's a pale beer. You have to add hops to get it to a bit of hops. Anyway, to the point. So what we have then is the calcium takes out the bicarbonate, reduces the buffer capacity, what they call residual alkalinity. And the magnesium is countering calcium because calcium makes insoluble salts, magnesium makes soluble salts, i.e. retain it in a solution. So that's one aspect. So you've got to get the pH right, but also you've got these flavor profiles which are added by the sulfate, gives a very dry flavor, the chlorides, which also can affect the flavor. So what you want to do is, although you might think, well, the negative, you extenuate them, the enhancement. And so, for example, sulfates enhance what? The malts, or does it enhance the hops? Well done. Yeah, because IPA is really high, and that's why it's the best bitter in the world, even though it's not. But anyway, so yeah, so you enhance the hop. So if you've got high sulfate, you're going to have a hoppy beer. The chlorides enhance the malts, a malty flavor. And like I said, if this was if we were back in normal times, we would kind of bring in. Well, I think I did bring in some hops, didn't I? Anyway, so you'll taste the difference between a hop and a malt. It's a very different thing. So you consider that, and this is what beer styles do. And as I mentioned to you, our ancestors didn't really understand any of this, but so they did it by trial and error. And even today, brewers like to do different things. And so the mixing, they actually define the water most of the time, define the sulfate, define the chloride. And as we talked about, on occasion where, for example, we've got very little calcium, but high bicarb, there's two things we can do. We can add dark malt, which is traditional for a stout, because the malt, dark malt has a lot of phytic acid. Phytic acid is phosphate, as you know. Phosphate is a buffer. So again, we can take out to regulate the pH based on that. Or if we wanted to make a Burton beer, we increase the calcium by adding gypsum, calcium sulfate. It has the calcium to knock out equivalent to the bicarb, and it's got the sulfate to bring out the hops, the bitterness. So that's the kind of thing you need to have, if you like. Where are we going next? Yeah, things like this, you know, a bit like we had in the midterm, one of the long answer questions was, I can't remember, to be honest. I think it's vinegar kombucha. What was the question on the midterm? Do you remember? I can't remember. The big long answer. Was it kombucha, was it? Yeah, so we're certain, and you can see these questions coming, I suppose, in a way, is that when you're given a question saying, explain the brewing process, I think what I noticed in the midterm, thinking back, is that, yeah, people drew the sort of different steps, which is fine, but they didn't go into real detail about what happened at each one of those steps. And what you have to do in an exam, and you've taken so many exams, that you know, I don't need to tell you this, is you put down anything you can. You know, you don't have to spend the next 3 hours there, but as you know, if you come into an exam, I never really keep to the timeline, in that, you know, I've been here 1.5 hours past the exam time, waiting for one person to finish. It's no problem, you know, because this is a time you can write down. But when you're doing, if there is a question on brewing and about the brewing process, it goes to reason. You would draw the flow diagram, and you would just put a little piece or a sentence or two saying, at this stage, X, Y. And then you might talk, and if a question asks, what's the quality metrics? Well, you can talk about, oh, we measure diacetyl, we measure alcohol content. I'm not suggesting this is going to be a question in the exam, but, you know, I think you get the idea. Yeah, with metabolic pathways, you know, I don't expect you to be able to draw out the Krebs cycle or anything like this. Or even with a glutamate, as I mentioned, you don't have to remember the sort of saying, oh yeah, the acetyl-CoA goes to citric acid, then round. You don't need to know pathways, but the principle of them. You know, the principle that, for example, when we're doing citric acid production, the malate being the anaplerotic reaction, and how it's got a antiport. You know, malate goes into the mitochondria, citric acid comes out, gets transported out. Other questions such as, or other information such as the fermentation system, which is obviously a Subner, or it could be a surface one in the older ones, and the conditions. And you know about citric acid production. One of the note things about it, low pH to knock out the glucose oxidase, and also limiting nitrogen and things like that. Yeah, so that's all we need to do. Yeah, so I could keep going on if you want. Do that. Not sure what I'll talk about next, but I'll think of something. All right then, yeah, wastewater, why not? All right then, can anyone tell me then, what's the difference between treating municipal water and treating wastewater? And when we say municipal water, we basically mean drinking water. Now, the reason why I bring this up is because when we do exams, well, we still do exams, people get confused. And I noticed some people got confused in the unit summary, thinking that municipal water treatment is the same as wastewater treatment. But the key difference is that with municipal water, you've got to start with good quality water. You can't start with sewage and hope for the best, even though that's the future, people say. So it doesn't contain a biological step. The biological steps in wastewater treatment is basically to remove nutrients from water, because if we don't remove those nutrients, when we put it into rivers, the blooms come, bog toxin comes, poor fishes die, and you get stagnant rivers, like the River Thames back in the 1800s, I suppose. So with I'm not presenting it again. So with municipal water treatment, we start off with fairly clean water. We have a reservoir, so it settles naturally. Then we add a coagulant, and we talked about how we use the jar test to know what coagulant to use and concentration. We can't add too much because the coagulant becomes an issue. And I think I told you about cases where there was excess alum in water, and that causes neurological issues. And if you were a cynic and conspiracy theory, you would say there's something going in Nova Scotia when they had all those 42 cases of people who suffered from neurological issues. They kind of were going on about the cyanobacterium toxin. And I said, oh no, there was no toxin there. But then you think, well, I wonder if it was something to do with the water, putting too much alum in. But public health or public health, we won't go into that. Get to the point. The point is that we use a coagulant to get all the insoluble, as much insoluble material as you can to make it soluble by agglomerating it. The flocculation tanks enhance that, but it also drives off any volatiles, which we know. And the sludge we collect isn't so much biological, it's basically coagulated. And then we can sand filter it, we can chlorinate it, and we take the sludge, which is just basic organic material, and put it onto land or use it for other purposes. But with wastewater treatment, we need that biological step to fix the nitrogen and to fix the phosphorus and to fix the carbon. And what we use as metrics in wastewater treatment is exactly that, the biological oxygen demand. This is the constituents that could be metabolized by microbes, depleting the oxygen, killing the poor fish. We use chemical oxygen demand, which is not, it's similar to COD, but it's a chemical oxidation, what can be oxidized, which again has the same effect, depleting the oxygen. A very loose measure is total soluble solids. These are solids which are soluble, as we talked about. And the volatile soluble, these are volatile ones are the worst ones in a way, because they add to the impact of the aroma of the water and things like that. But on another side, they're very good to remove because they're volatile, so they kind of go away. And the turbidity, people have got varying acceptability for turbidity. There's some water you get, I think it's in Bury or somewhere, very turbid, but people drink it, say, oh yeah, it's what it is. Turbidity is a measure of colloidal, and it's not so good to have, but it's a measure. And what we do in terms of water treatment then is we've got to try and get all the pollutants which would to negatively affect river aqua life. And obviously, because water's basically circulated, what we're wasting one day, we're drinking probably a few days later. It's the way the world works. We need to make sure it is potable. And we do this by the activated sludge process, which is essentially there to drive off the volatiles, to make the biomass, to fix the nitrogen, to fix the phosphate, where we get sludge. And this sludge, we can't really dispose of it directly for the simple reason there'd be too much of it. So what we do is do an anaerobic digestion, and we know about Pasteur effects. Pasteur say, well, it's anaerobic, it's less biomass per unit substrate. And we produce the ammonia, the methane, and the methane is used as a energy source when it's cleaned up. And I say we can't use it directly for the simple reason it's got moisture in. So it's actually quite an effort to strip it of that moisture in order to use it. But people are saying, oh yeah, we're going to have biogas and that. Everyone's going to have a biodigester in the backyard. The problem is, and they're going to find this out, it's all about having these biodigesters, but you've got to keep them fed. And it's interesting, in France, I think it was, where they've really gone all in biodigesters. They actually have to start using food, well, crops, should I say, in order to keep the fermenters going. So basically, this concept saying, oh, we'll get the waste, we biodigest it, but then they're using good crops to feed the biodigester. So, it's one of those kind of contemplation things, isn't it? But the important thing is, anyway, I'm going on too much, is that you've got a biological step in the wastewater. There's a difference between the two. And this was a small thing with the sort of unit summaries that people said, oh yeah, in the anaerobic digest, we have these 3 steps. One where we're breaking down proteins, lipids, and things like that, and then we make acidogenic bacteria, and they make the acetic acid, which then gets converted to methane. It's good just to know don't need no specific bacterial names, but Clostridia, Methanogen, things like, because it is a micro course at the end of the day, isn't it? And I suppose thinking about biofuels, one of the big questions, well, not big, some questions that get asked is about what you need to remember the butanol pathway. And the butanol pathway, I think it's one of the more complex we ever come across. Although it's not, like I say, it's not complex when you think about I'm just going to get it for you. It's not that complex when you think about how it operates, but that's basically what you have to. If I like to say, I don't know what questions are going to ask. It's like, but if you're going to ask a question on biobutanol production, I think it'll be a very big, high expectation to say, well, draw out the acetone, butanol, ethanol pathway of Clostridia and tell me how it works. I think that would be a bit too mean, to be honest, but I'll just get it for you. But what you can do with, I know I'm repeating a bit now, but you've just got to show the principle of it. The principle of why it does this acidogenic phase first, and then follows through with a solvent phase. So as we talked about, as Clostridia grow, because they're obligate anaerobes, they can't use electron transport chains, but they've got something similar called the ferredoxin. So ferredoxin, it spans the membrane, it's electron transfer, and it metabolizes to pyruvate, as evolution has told us. That's quite universal, not exclusive. And from pyruvate, it transfers the electrons to the ferredoxin, which then spans the membrane, pumps out protons, and it also has a hydrogenase, which produces hydrogen, which is, people say, oh yeah, we'll use hydrogen, isn't it? And as it's growing, it needs ATP. So we get the acids produced, as we talked about. But then when it hits, say, pH base, if you want to call it that, it's too acidic. It needs to start sporulating and things like that. So it has to increase the pH. It can't go any further. So it kind of puts things in reverse. And when it puts things in reverse, it's assimilating the acids to fight off the acidity, because you can imagine, it wants to get rid of the acids now. And it does this by producing butanol, ethanol, and acetone. And the key thing to remember is the rate limiting step is actually the NADPH or NADPH generation. Because it goes that way. Well, it's not. So you're generating NADPH for the final step here, and that's the rate limiting enzyme. And NADPH typically is an anabolic redox carrier. So NAD is for catabolic reactions, NADPH is for anabolic, building up. And in this case, so once it hits the stationary phase, it kind of reverses everything. So it's no longer starting to regenerate NAD, but it's actually producing NADPH. And it does that, and it then stimulates the acids to produce the solvent. And it could also at that time produce, and it's also got these granules, which are, you wouldn't need to know that because it's superfluous. And so if you're asking a question of this kind, you kind of would be asking the general idea. You don't need to make the pathway, but you can say, well, it produces these and this and that, and it re-stimulates to produce the solvent. And you might put a profile in, but that's all you need to do. You don't need to, but like I said, I don't think I've ever asked, did I? I can't remember if I asked the question or not, to be honest, but there you are. All right. Anything else burning or? Try and find something else. All right. So in food fermentations, obviously what we have is more of an array, and there's so many different fermented foods. You can't answer all of them, and like I would never ask saying, that picture I showed about the different microbes, the yeast and the bacteria involved in food fermentation, if you could recreate that and answer all the names, you don't need to do that at all. But for the food fermentation, you've got to have a sort of grasp on the basics. I know I'm going probably kind of going over it again, but you don't know to know each step per se in detail, but the general idea about what the fermentation is. And when we talk about cultures, the concept of domestication, as we've covered quite a few times. And when would you use spontaneous fermentation? When would you use starter cultures? When would you use backstopping? So that's actually a question I could ask now. So what do you think is the defining decision you make when you're actually saying, well, I'm going to be Klaus down at the Reif Winery. And if you remember, when we visited him, they actually initially started making wine from spontaneous fermentation. But they kind of went against it to start a starter culture. But what is the decision making that goes into that? You can answer online as well if you're still on.
If you can use a pure culture, you generally use like a single strain. You'll generally use a starter. I guess if it's or they call it succession. This succession, you wouldn't backslop because you lose the first microbe. And spontaneous is
Exactly right. So, what starter cultures give you is consistency. You don't have to say, God, I hope the yeast is there. It also enables you to inoculate an active culture, which allow compete competitors. And as you rightly said, when you look at what we use in spontaneous fermentations, these are the ones that have successions. And you could argue saying, well, there's a few fermentations where we do black stopping and they've got successions. But when we look at the ones that are spontaneous, you do it for two reasons. One, well, you want to say, look, I'm a pure winemaker and I only depend on the yeast, like my ancestors depended on the yeast and the grapes. But with sauerkraut, that is a succession. So if you had a starter culture, and it doesn't stop people trying to make them for definite, is that the whole point is the microbes are going to be on your product anyway. So why add to them? But if you took end of sauerkraut and just took the end, you'll probably have a lot of Lactobacillus plantarum, but not much leuconostoc. And as we talked about in the sauerkraut fermentation, it's all based on the leuconostoc starting off a reaction. If the leuconostocs aren't there, you're going to have a big issue of getting the acidity to get plantarum work, and you'll probably get invaded by contaminants, Awinia, Pectobacterium, which will make your cabbage all spoiled, wilting, and things like that. So spontaneous fermentations, tradition, yes, it's but a lot of the time it's to do with that succession. And if you think about kefir grains as well, in a way, that's not spontaneous because it's a bit like when we're making vinegar, or well, not, vinegar's a good example, I suppose, as is kombucha. You're not having a succession there. It's more like a coordination. Now, that coordination in the case of kombucha is a bit antagonistic. The yeast producing all this ethanol, upsetting the acetic acid bacteria. Get back, so I'm going to produce a lot of acetic acid to keep you backwards. But that's the decisions you make. And in food fermentation, as we talked about the GMO, a lot of the time you're just trying to decide. We like tradition. We domesticate our microbes in order to do that. But even when you get GMO, people going against it, when you think the reality is when people get onto things, as you probably learned in the pandemic, they get on with them without any notion about logic and whatnot. There's no reason why GMO would be bad in fermented foods. And you could imagine you'll get so much consistency with it. And there's benefits to it, but it's all harking back to saying, well, we can't do this, we can't do that. And I suppose it's the journeys onto the unknown, and a lot of the innovations in the world of in science especially have come with that. And the pandemic, if you like, has really underlined it. When we think about all these technologies we have in terms of ventilation, mass technology even, people in the pandemic just went back to the 1918 playbook where they were kind of wearing cloth masks saying, this works, or wiping down the packaging. Anyway, I won't go on about that, but anything new is always hard. In the food industry, you get much more, and a bit like the microbrewers, you get much more traction by being small, traditional, than you would be at a bat. And, it's very interesting with the beer, as we talked about, it's dropping 10% each year, the market, as we kind of switch on to wine. And when you think about the wine and brewing, you can see the difference between them. Winemaking is small. You don't know what you're going to get one year to the next. The style, but it's got stories to tell. With brewing, it's mass manufacture, consolidation. It was interesting, Carl saying he's going to open a brewery at his wine place. I thought that was quite ironic, but yeah, this is what the food fermentations do. But anyway, to the point, before I forget, when we look at food fermentation, again, have the basic concepts of them. And, when we talk about successions, at least know what the successions are and things like that. But anyway, so any last questions? Because I think we're running out of time. No? All right. Well, good luck with that, and get back if you need anything. And like I say, I will release the answers to the quiz if everyone's answered. I will have to shut the quiz down for that to do that, for the obvious reason. but I'll try and get them up. And the exam, as I say, it's going to be on the 21st, I think it is. Take-home exam's going to be on the 18th. Well, the information is going to be released on the 18th.